that obviously is always subject. Last time we finished, we started into chapter 21, okay? We talked about the non-specific defenses, the essentially external and internal non-specific defenses that are kick in every time that we have an invasion of some type, okay? Now we're going to talk about the adaptive defenses, which are more specific or specific antigens. So an example of this would be the COVID-19, the antigens are antigens associated with the virus. Remember, there's not a single antigen on the virus. There's not a single antigen on anything. So one of the things in developing a vaccine, they have to find a vaccine that's going to be specific against specific antigen, and that's going to give rise to, hopefully give rise to immunity, okay? So we have to have an exposure to an invader, okay? And results in a specific program response and a memory of the pathogen, okay? It is specific antigen, specific response. It is systematic. In our previous discussions about the non-specific defenses, you know, if you had an invasion on your left forearm, you had a breach of the barrier, breach of the skin on your left forearm, that is where the fight was going on. That's where the battle was going on, okay? But in this, it's a systemic response. It has memory in each one of the types of cellular immunity and humoral immunity or antibody immunity. Both of these types of cells, one of the lines of the cells is a memory cell, okay? So that if you have this same type of infection again, then you will have the ability to mount a response, mount a greater response, and mount a faster response. And then it's based on antigen recognition, okay? Always remember that we have antigens. Each one of us sitting here listening to this lecture, and each person that you see, okay, unless they're an identical twin, has their own self-antigens, The immune system is able to recognize that unless there's a clinical abnormality, unless there's a pathological condition, and it's able to recognize that, and because it recognizes it as self, there is no response mounted to self, okay? It is when an antigen is recognized as non-self. In other words, not part of me, not part of you, not part of every individual you see, okay? That is when a response is mounted on non-self antigens, okay? An antigen is a foreign substance that elicits an immune response. Major histocompatibility complex 1, okay? That should be a 1, it looks like an I to me, but that's MHC1. There's MHC1 and MHC2. MHC1 is on every cell in our body except for red blood cells. It's on every cell in our body except for red blood cells. And it usually displays in a normal non-pathological condition, displays self-antigens. In a normal non-pathological condition, displays self-antigens. So, if that's somebody on this microphone's not muted, please mute your microphone. I will mute it for you. And so displays self-antigens. And how does it get self-antigens? As part of the breakdown process, some of that material is transferred into the into the endoplasmic reticulum, and is packaged in the Golgi apparatus, taken up to the plasma membrane, and those self-antigens are displayed. And so that's major histocompatibility complex 1. Healthy cells display just self-antigens. Infected cells also bind fragments of foreign antigen from within. So these are cells that have become infected, okay? Cells that have become infected with bacteria or viruses. That bacteria or virus has been broken down, that antigen has been attached to a protein, to a plasma protein, and has been carried up to Golgi by way of the Golgi apparatus up to the plasma membrane and is displayed as non-self antigen, okay, as a foreign antigen. So normally, major histocompatibility complex 1 displays self-antigens in healthy cells. Major histocompatibility complex 2 is only on the macrophages and dendritic cells or cells of Langerhans. Those are the immune-like cells, macrophage-like, immune-like cells at the junction between the epidermis and the dermis that we learned about in chapter 5. We just went over it in our anatomy and physiology complex, okay? And this is a particular thing that's going to display on the macrophages and the dendritic cells, and it's going to present that to the CD4. We'll learn about their CD4 and CD8 T cells, okay? This is going to display this to the CD4 cells, okay? So major histocompatibility complex 1, okay, displays self-antigens on every cell in our body except red blood cells. Major histocompatibility complex 2 is only on two particular types of cells, dendritic cells and macrophages, and it's going to display a particular type of non-self antigens. It doesn't display self antigens, it's only non-self antigens. The antigens are bigger. It's trying to elicit a response from the CD4 cells. We'll get into that into a little bit more detail. There's 2 types of responses. Number 1, a humoral or an antibody response, okay? So we get an antibody response from B cells. How I remember that? Antibody's got to be in it, B's got to be in it, okay? Obviously, if you have an old book test, you don't have to remember things like that. But if you remember these little things that I tell you, 2 or 3 years from now, 4 years from now, 5 years from now, when you're taking your boards or whatever you're taking, they pop one of these questions in there that you didn't know that was going to be in there. You can remember the antibodies got to be in it and B cells got to be on it, and they're the ones that produce antibodies, okay? Our T cell immunity, okay? Cell-mediated immunity. So these are the 2 types of specific directed responses, okay? And they're not completely independent of each other. You can have an antigen presented and you can have a cellular response and an antibody response. Cellular response is going to be more immediate, okay? Cellular response is going to be more immediate. The antibody response is going to be, it's going to take a longer time to go on. Some of the problems in the pathology in the lungs in the COVID-19 infection is related to the cellular response, okay? So related to the overactivity of the cellular response. So after you go through this chapter and you get an opportunity, don't read it on Facebook. Go to the paper, go to YouTube. There's a good virologist out of Columbia University. He's got a whole set of virology lectures that he gives to his students, and he also has updates on the COVID-19 virus. You can learn a lot of information just by watching YouTube videos. I learned a lot of things just on YouTube video, and if I can impart anything to you, you can learn anything you want to. You can learn anything you want to on the internet, okay? There are a ton of resources for learning whatever you want to, either typing in Google or going to YouTube. And so if there's some fact that you want to learn or some concept, there's a lot of things I don't know how to do on Canvas, and I just go in and type how to do this on Canvas. And I learn how to do it, and I'm able to do things. 3 major types of cells. We have B cells, okay? B cells, primarily antibody-producing cells. T cells, cellular response. And antigen-presenting cells. Antigen-presenting cells. That's what APC stands for. Antigen-presenting cells. Antigen-presenting cells are cells that obviously present antigens. They're the dendritic cells, the cells of Langerhans, okay? The dendritic cells of Langerhans. In anatomy and physiology, when I was in medical school, everything had not everything, but 90% of stuff had people's names attached to it. We're getting away from that, okay? The only kicker in this is you and I think about Dendrites is things attached to nerves, okay? Well, they call these dendritic cells because they look like they have dendrite attachments, okay? Those are what is accepting signals from receptors or from other nerves is the dendrites on the nerve cell, okay? But it's not actually a nerve cell, it's an immune cell. And they're also called Langerhans and B lymphocytes. These engulf organisms and present foreign antigens in their own plasma membrane, okay? As opposed to the MHC1, if they are presenting a foreign antigen, that cell has become infected, okay? That cell has become infected. These cells, the antigen presenting cells, actually engulf these whatever these foreign materials are, with one of the purposes of being able to present this foreign antigen on its own plasma membrane, okay? And this is to elicit response from the B cells and also from the T cells. Don't think of cellular immunity and humoral immunity, antibody immunity as completely separate from each other. They actually interact. The helper T cells, which is what the stimulation of the CD4 cells results in, is helper T cells. They actually work on both sides. They work on the antibody side and they work on the cellular side. And then finally, another type of cell is the macrophages, as we've learned previously. So these are antigen presenting cells, okay? These are antigen presenting cells, primarily the dendritic cells and the macrophages that are actually going to engulf the foreign material, digest it down, take that digestive material into the endoplasmic reticulum, have it packaged up in the Golgi apparatus, taken up to the plasma membrane, and presented as an antigen, okay? B cell response, antibody-mediated response. What does the B cells do? Antibodies are released, as you can imagine, okay? Well, in order to have an antibody released, we have to have an exposure, okay? B cells are produced in the red bone marrow and mature in the red bone marrow. B cells are produced in the red bone marrow and mature in the red bone marrow. They have a receptor for a certain antigen. They have a receptor for a certain antigen. May have upwards of 1 million different B cells of the body waiting around for these specific antigens. This is the only downside about having this on the computer screen and not on the big screen. All right, we have a primary response where we have an antigen here. Okay, we have an antigen. They encounter the B cells. Okay, they encounter the B cells. The B cells have a specific receptor for this antigen. So this antigen fits into this specific receptor. Okay, once this antigen fits into the specific receptor, this activates the B cells. This activates the B cells. Once these B cells are activated, the B cells are converted to plasma cells. These are the effector B cells. Effector B cells means it's going to carry out some type of work. Okay, it's going to do something. Okay, so once a specific antigen meets a specific receptor on a B cell. Okay, once it meets a specific receptor on a B cell. Okay, notice that these particular antigens don't fit on these. These are teal receptors. These are blue receptors, I assume. These are specific for these receptors, but not for these receptors. So this particular B cell is not activated, whereas these particular B cells are activated. We're going to activate our B cells. Then we're going to form a group of plasma cells, okay, clones of plasma cells, and a memory B cell, a memory B cell. These plasma cells are going to secrete antibodies. So this is our primary response. We are exposed to an antigen. Okay, that antigen could be an immunization we got when we were young. Okay, could be an immunization we got when we were young with an attenuated antigen. Okay, we got exposed to that antigen. We had an interaction with one of our B cells. These were activated. We developed plasma cells. Okay, that secreted some antibodies and memory cells. Obviously, if we had an injection, with an immunization, there wasn't an active infection going on. So what few antibodies were produced interacted with that attenuated antigen, okay, and then that response was over. That response was over. But we had these memory cells here. So the whole purpose of that is that if some point, okay, if some point later on, and I'll use the example of the polio vaccine, okay? I told you that we got the polio vaccine, the Sabin vaccine on a sugar cube, okay? When I was, my dad took us to high school. That was when I was early growing up until I think in the, just before I turned a teenager, there was no vaccine for polio, and people were getting polio, and they were polio hospitals where you went to, and they put you in an iron lung because if you got your respiratory muscles paralyzed, you couldn't breathe, and all the iron lung did was change the pressure inside the chamber. Okay, if you change the pressure inside the chamber, the pressure effect on your chest wall caused the chest wall to expand, and air went into your lungs. Well, once we got that polio vaccine on that sugar cube, and I think they may have been 1, 2, or 3 steps, I can't remember, but then now we had memory cells. So I would have contacted, and if you've been immunized against polio, you have come in contact with the polio virus, okay? But because either we did, it didn't develop into an active infection, or if it attempted to develop into an active infection, we had memory B cells, and the memory B cells elicited the response. And once they elicited the response, then we were able to fight this infection, mount the infection right away, And in 48 hours, we've mounted the infection. And then we make huge amounts of cloned plasma cells, that are secreting antibodies. And so that's how an antibody reaction works. And then for the people that are making antibodies to the COVID-19 virus, this is what's happening here, okay? When the B cell is triggered by the antigen, it divides and each matures into a plasma cell. Plasma cells produce approximately 2,000 antibodies a second. Can also be triggered by T cell secretions. Okay, so if T cells have specific secretions, that also can trigger this response. Effective against extracellular pathogens such as bacteria, viruses, and toxins, and antigens and body fluids. Okay, so the humoral response is effective against extracellular pathogens such as bacteria, viruses, and toxins, and antigens and body fluids. So if we got a vaccination, that's what we would get. We would have it injected into our arm or our thigh, it depends on how young we are. And so those antigens would be in body fluids. Here's what happens when you have, there's a number of possibilities that can happen if you have this antigen antibody complex. Number one, it can neutralize the antigen. Okay, it can neutralize the antigen. It can cause an agglutination, okay? So you can either neutralize it, it can cause an agglutination. In other words, it can bind a bunch of these antigens up and make them ineffective, or it can cause a precipitation. That is one particular chain, okay? The other chain, remember we talked about complement. What complement does is actually put a hole in the plasma membrane. It puts a canal in the plasma membrane, an open channel, and essentially once you do that, the cell is going to rupture because the cell is highly osmolar. It has a high osmolarity, and because of that, and the extracellular fluid has a low osmolarity, so water is going to pour in from outside into the cell, and the cell is going to rupture, okay, or become non-functional. So one path we can go down, we can neutralize it, we can agglutinate it, or we can precipitate it. And if that happens, then we either get phagocytosis by the macrophages, we can get inflammation, okay, we get the inflammatory response, or we can get cell lysis. So this is, once we have this antigen antibody complex reaction, what happens then? These are the four possible scenarios that happen. We can neutralize it, we can agglutinate it, we can precipitate it, we can put a hole in the membrane. Then we either get it phagocytized, in other words, a macrophage comes by and eats it, digests it. We can have an inflammatory response, and neutrophils come by and take care of it, or the cell can be lysed. And so that's what happens in an antigen antibody complex reaction. What does an antibody look like? An antibody has two chains. It has two heavy chains and two light chains. It has two heavy chains and two light chains. The heavy chains have, and the light chains both have a constant region, constant region. This is the light chain, this is the heavy chain. This is the light chain, this is the heavy chain. Light chain, heavy chain, light chain, heavy chain. Heavy chain, heavy chain. The longer ones are the heavy chains. These are light chains, light chains. The brighter area is the variable portion of the antibody. The lighter area is the constant portion of the antibody. The variable portion of the antibody is what determines the specific antigen it's going to react with. So we have a basic building block, okay? Just like a basic building block of a car. We've got a chassis, we've got the wheels and chassis, and we put a body on it, and then we can do whatever we want to with it, okay? Well, it's just sort of the same thing. We've got the basic build. It requires four chains, two heavy and two light, constant regions, which mean they're all similar, okay? In other words, this antibody is similar to another type of antibody, which is similar to another type of antibody, except for the variable regions. And the variable regions determine what specific antigens are there, okay? Two arms, one heavy, one short. Constant regions, variable regions where the amino acid sequence is different, therefore creating the antigen antibody reaction. They're creating the different antibodies, okay? They are different classes of antibodies. You can look at this picture if you want to, but we're just going to talk about the classes. IgG is far and away the most common, okay? When people are getting plasma transfers, when the COVID-19 recovered patients are donating plasma and transferring plasma to patients that are having active COVID-19 infections, this is the antibody being transferred, the IgG, okay? This is what antibody is. It's not the initial response. The IgM is actually an initial response to an antigen antibody reaction, but the IgG is the one that provides the protection. So this is the antibody being transferred from somebody who's recovered from COVID-19 to a patient who has an active COVID-19 disease. And so this is one of the types of treatment that's being tried. Remember, we have no effective treatments and we have no vaccine now. We have no effective treatments and no vaccine for the treatment of COVID-19 right now. Encourages phagocytosis, triggers complement activation, antibacterial, antiviral, antitoxin. Essentially, an immunization is a toxin injected into the body, okay? An immunization or a deactivated virus. Is a toxin or deactivated virus, okay? It's also antibacterial. Complement activity, remember, complement is where it puts a hole in the membrane. That's what it's essentially what the complement system does. This is the largest percent of antibodies in the human body, okay? IgA is another class of antibodies that's primarily in milk and milk secretions, it's found in secretions. It stops the pathogens from attaching to the epithelial cells. So if a pathogen comes in, by the respiratory tract, oral tract, and there's IgA present, okay? Or if you get secretions, either milk and colostrum, okay, this IgA will stop the pathogens from attacking the epithelial cells and spreading, okay? We also have IgM, okay? IgM is responsible primarily in the blood incompatibility reactions. It's a less percentage thing. The primary two ones that we concentrate on are IgM and IgG. It's primarily IgG, but this is bound to the beta lymphocytes. It's activates complement, and it's the one that if you mismatch a blood typing this would be the one that would be involved, okay? And then IgE is the one that, this is the one that results in the hypersensitivity reactions. If somebody is allergic to penicillin, okay, they get their first dose of penicillin, they get exposed to it, they get a second dose of penicillin, okay, and they go in anaphylactic shock. Or they have a severe reaction. My youngest brother is allergic to penicillin, and we were coming home, he got his dose of penicillin. This probably been the second time. He just started throwing up in the car. And we just turned around. My mom just turned around and went back to the hospital. My niece, my first cousin, she was a pharmacy tech, and she was so allergic to penicillin that she couldn't work in the pharmacy because just being in the presence of penicillin would cause her to have these allergies. But IgG is the one that attaches to the mast cells and responsible for the severe. It's also present in parasitic infection, but it's responsible for the severe reactions to a particular substance. So a particular antigen would result in IgG being performed, and IgG would attach to mast cells, okay, and would cause a release of histamine, and there's a number of problems associated with that. We'll talk about it, okay. So we've talked about humoral immunity. Now we're going to talk about cell-mediated immunity. Cell-mediated immunity is a little bit more complex, okay? It's a little bit more complex. We're not going to delve into it. Obviously, it's very, very complex. You can get your doctorate in immunology. You can take several graduate courses in immunology. You can take undergraduate courses in immunology. We're just doing a survey on cellular immunity, okay? So don't get too bogged down. This is an example. You read them, you look at the slides, do the student notes. If there are a number of videos on YouTube, you just type in cellular immunity, watch a video or two. Some of them will be 5 minutes, some of them will be 50 minutes, and you can go into as much depth as you want to. But this is a cellular attack, okay? The antibody attack was specific antibodies. The cells, other than being presented to the antigen, never really interacted with the actual invaders, okay? So an antibody interacts with the invaders, but the B cells and the plasma cells and the memory cells did not interact with the invaders. They were only exposed to the antigen, and because of exposure to the antigen, they produced antibodies. These particular cells, especially the killer T cells, actually go up and fight with the cells, go to battle with the cells that have the foreign material in it. Okay, T cells are produced in the thymus gland. Okay, so B cells are produced in the bone marrow. T cells are produced in the thymus gland. Actually, that's where they mature. They mature in the thymus gland. They directly attack the invading antigen by going after the infected cell. So this is the difference between T cells and B cells. B cells don't directly attack the infected cell. They just interact with the antigen, okay? Effective against intercellular pathogens such as fungi, parasites, viruses, cancer cells, foreign tissue transplants. So when a person has a kidney transplant and it is rejected, it is a cellular response. It's a cellular response. People who have transplants, there again, I haven't been in medicine in a number of years, but used to have to take a lot of medication to decrease their immune response, because unless they got tissue from an identical twin, that tissue was non-self. Unless they got a kidney from an identical twin, that tissue was non-self. So they were going to mount a immune response. So they had to take a number of immune suppression drugs. And you can imagine this would set them up for the possibility of another type of infection, okay, or another type of invasion. So the process starts with activation of some T cells by a foreign invader, okay? So we have a dendritic cell, okay? We have a dendritic cell here that is displaying It's an MHC2. It's an MHC2. It is displaying a processed bacterial antigen and also has a co-stimulatory molecule. You have to remember this is a two-step process. It's a two-step process. So you're going to have to have a co-stimulating molecule and some other process that it undergoes for the CD4 cells. These are CD4 cells that are going to interact. So there's going to have to be some kind of double recognition. So the dendritic cell engulfs the antigen, processes it, and displays a fragment on the MHC2, this major histocompatibility complex protein. Okay, this fragment is going to be a longer fragment. It's going to be a longer fragment than a foreign fragment displayed by the MHC1. Normally, MHC1 just displays self-antigens. The non-impacted cell, MHC1, just displays self-antigens. Remember, these are cells that have actually engulfed, okay? They have engulfed an antigen, okay? The MHC1, if they display a foreign antigen, they have become infected. These actually go out and look for foreign and engulf it and then display the antigen, okay? The CD4 cells recognize the antigen, the complex, okay? And then there's some other T cell receptor response, and the CD4 proteins bind to this thing. And then there's a co-stimulatory molecule. I'm not so interested in you knowing 2A and 2B as you know that there's some kind of double recognition thing. The reason there's double recognition is it's a fail-safe mechanism. Like on an airplane, there's there's a backup, okay? You might have a steering mechanism, but that goes out, there's a backup for it. This is the same type of thing. It requires a double recognition to start this process, because if it only had a single recognition, okay, and it made a mistake, if it only had a single recognition and made a mistake, then it is going to produce a clone line. It's going to produce a clone line. So see down here in step 3, we've got clone activation, okay, of helper T cells and memory CD4 T cells, okay. It's going to start a clone line, and essentially that clone line is going to attack your own cells, okay. It's going to attack your own cells. So this is a particular, and I'm using this, I'm describing a scenario that may or may not be set up by this, but this is one of the theories as to why there's a double recognition. Like I say, I'm not interested in you knowing 2A and 2B. I'm interested in you knowing that there's a double recognition system. The antigens presented on the class 2 MHC, major histocompatibility complex, okay? This antigen was engulfed, okay? It was engulfed by dendritic cell or macrophage. There is a double recognition system. The reason the double recognition system is so the system doesn't make a mistake. And because of this double recognition, we now have clone formation, okay? We now have memory CD4 cells, and we have helper T cells, okay? So in reaction to this, okay, with the CD4 cells, these are CD4 cells. Remember, on the T cells, we have CD4 and CD8, okay? The CD4 cells are going to go into CD4 memory cells, okay, and CD4 helper T cells. So what I'm interested in this picture is antigen recognition, knowing that this is a dendritic cell or macrophage, knowing it's MHC2, okay? The antigens presented, there's a double recognition, then a clone formation of the CD4 cells, and they are memory CD4 cells and helper CD4 cells. That is what I'm looking for you to remember from this slide. Foreign antigens presented by the antigen presenting cell. This dendritic cell is an antigen presenting cell, okay? Antigen is recognized and binds to the T cell, as well as an endogenous antigen from invader or from cell as a signal to the T cell, okay? In the second step, a co-stimulator must be present. It can either be a cytokine, plasma membrane molecule. So I'm not so much interested that you know what the individual 2 steps are, as I've said about 4 times now. I'm interested that you know there's a double recognition thing. So the immune response, probably 2-step, is so the immune response is not set off accidentally. because if it gets set off, then you're gonna have a line of clone cells. Okay, so this gives rise to T cells, which differentiate. And so let's look at this diagram here. So these are our CD4 cells that have been activated. Okay, these activation results in memory cells. So we have a CD4 memory cell and effector cells, which are the helper cells, We then have similar activation here with the CD8 cells, okay, with the CD8 cells. Except with the CD8 cells, this is class 1 major histocompatibility complex, okay? This is also an antigen presenting cell, okay? And we're going to have a formation of a CD8 memory cell, and we're going to have CD8 cytotoxic or killer T cells, okay? So if you want to have a good idea about the cellular immunity, how it works, what activates it, okay? What activates the CD4 cells, what activates the CD8 cells, what is the result of the activation of the CD4 cells and CD8 cells. CD8 has a CD8 memory cell, CD4 has a CD4 memory cell. CD4 has helper T cells. These are helper T cells. They're called the effector cells because they do the work. CD4 has the helper T cells. CD8 has the cytotoxic T cells, the killer T cells, okay? So this slide is a good slide. It's a really good slide of what goes on in the cellular immunity. Elimination of the intruder, okay? Types of T cells, cytotoxic T cells, the killer T cells, CD8 cells. So this is a cytotoxic cell attaching to a cancer cell, okay? Helper T cells, which are part of the CD4 cells. We have the regulatory cells. What the regulatory cells do, are once the process has ended, they turn the process off, okay? In other words, once the battle has been fought, then they turn the process off, and then we have memory cells, okay? So the CD8 cells go into a CD8 cytotoxic killer cell and CD8 memory cell. The CD4 cells go into helper T cells, regulatory cells, and memory cells. Helper T cells stimulate IL-2, that's interleukin-2, co-stimulator enhances proliferation. I'm not so much interested in you remembering the specifics here, okay? What they do is they help on both sides. They help on the cellular immunity side, the cellular response side, and the antibody side. So this is what I was talking about. There's the interaction between the cellular side and the humoral side, the antibody side, and it's by way of the CD4 helper T cells. Okay, they assist on both sides. And this is a positive feedback mechanism. Chapter 1 of Anatomy and Physiology, we talked about negative and positive feedback, and we'll talk about it later on when we get into this, some of the other systems. An example of a negative feedback is if your blood sugar goes up, you secrete insulin, it brings your blood sugar go down. If your blood sugar goes down, you secrete glucagon, your blood sugar goes back up. So it's trying to bring it into the opposite direction. A positive feedback mechanism is the clotting mechanism, the inflammatory response. Once it starts, it keeps going till it ends. Or we'll learn in reproduction and delivery, oxytocin, which is secreted by the posterior pituitary. Remember, as a tip, there's the hormone secreted by the pituitary. There's a bunch secreted by the pituitary, mostly by the anterior pituitary, but there's only 2 secreted by the posterior pituitary. Oxytocin, antidiuretic hormone. You learn those 2, and then if it's not those 2, then it's secreted by the anterior pituitary. That's how I remember those. But a positive feedback mechanism is the clotting mechanism that once clotting starts, it's going to continue to keep on going and keep on going and keep on going until that clot's formed, okay? It's not going to lessen, it's just going to, you're going to get more and more and more material. Unfortunately, there is a mechanism where where this an abnormal pathological condition where this keeps on, and you end up getting clots in your fingers and your toes and your extremities, and these people can end up with multiple amputations because it's an abnormal pathological condition we're not really sure, at the time we're not really sure about. So CD8 cells, okay, they recognize MHC1 class antigens, virus-infected cells, some tumor cells, and transplant tissue. When the antigens recognize, the T cell binds to the presenting macrophage and releases interleukin-1, becomes cytolytic. In other words, they go in and destroy the cell's DNA. Regulatory C cells stop the response when appropriate. Okay, most T cells undergo apoptosis, that's pre-programmed death. Okay, so other than the memory cells are going to hang around. The others have in other words, a pre-programmed lifespan, and they're going to after they're no longer needed, whether they're helper T cells or cytotoxic T cells, they're going to go ahead and die. Whereas the memory cells are going to go be around, and they're going to patrol the entire body looking for that specific antigen that they remember. Okay, can rapidly respond to reinfection. Some B cells do this also. Normally, even immature T cells are destroyed in the thymus. Okay, and so this is just a schematic. I'm a big fan of schematics. I'm a big fan of schematics because a lot of the stuff, this is a perfect example. I lecture on this, once a semester, twice a semester, depending on how many classes of anatomy and physiology too. May not have any that semester. And so this is always a chapter I come back to, because for me, this is one of the things, I never was an immunologist, I was a plastic surgeon, and it was never anything that I dealt with. Obviously patients had infections and had cancer, but it wasn't anything I dealt with. So I always had to come back and remind myself of a lot of this information. These schematics and tables, once you've learned the information, now if you don't know the information, they don't make any sense, although you can go to the picture and look at the picture and then go back and do the reading. Okay, you can go look at the picture and go back and do the reading. This is a good thing because it talks about the adaptive defenses and the innate defenses. Innate defenses is what we talked about last time, the surface barriers and the internal defenses. And then it talks about the particular cell lines we have here. These are the plasma cells, so this would be the humoral response. Notice that the helper T cells are interacting. They're interacting over here in the antibody production side. Okay, they're interacting in the antibody production side. And notice the CD4 cells are going to form memory cells and helper T cells. The CD8 cells are going to form memory cells, CD8 memory cells, and cytotoxic T cells. Okay, and eventually the whole process, either on the antibody side, is to produce antibodies, or on the cell side is either engulfed by the macrophages, inactivation, agglutination, put a hole in the membrane, neutralization, whatever needs to be done so that they eventually can be deactivated. So this is a good summary slide. Okay, good summary slide. So when I was taking my next test, I might have, if you've got a physical book, I personally don't like ebooks, but it seems like every course I take, you get an ebook now. I like having physical books where I can put stickers and things, but that's what goes on now. Memory, okay. Memory will remain after your first exposure. You have to have an exposure, okay? You have to have an exposure. Memory will remain after your first exposure. Antibody titers measure levels in the serum. That is the question now. We can measure antibody titers for the COVID-19, but we don't know if what particular level of titer, okay, what particular level of titer imparts immunity, okay? We don't even know if the particular antibodies being formed impart immunity. There are some reports of people being reinfected with the COVID-19 virus. And so it's one of two things. The one thing it could be, to say you're no longer infected, you have to have 2 negative tests. Well, the tests have certain levels of sensitivity. So you may actually still have a subclinical infection that is no longer picked up by the test, okay? because the level of virus, the level of infection in your body is so small that the sensitivity to the test doesn't pick it up, or the antibodies produced, okay, didn't impart immunity or didn't impart immunity right away, which means that we're not getting the antibodies produced to a specific antigen, or the virus could have mutated. Okay, viruses mutate. If you ever want to learn anything, or if you ever, probably right now, if you have an interest in being a virologist, there'd be a wide open field. You could get an MD, PhD in virology, okay, and just, the world will be your oyster. This is our initial primary response, okay? This is our initial primary response to a particular antigen, okay? This is our initial primary response to a particular antigen. These are antibodies being produced. At about day 10, we get a production of antibodies, but it's not a huge production, okay? Because we are being stimulated by limited antigen, okay? We get stimulated by limited antigen. So this would be our immunization, okay? This would be our immunization against mumps, diphtheria, whooping cough, pertussis, whooping cough, whatever the things you get, and we would develop an antibody response, okay? And then here's an example. Now we're exposed to two different antigens. Antigen A, which we've already been exposed to, and then a new antigen B, okay, the new antigen B essentially gets the same delayed response, but the second time we've been exposed to antigen A, we get an almost immediate response, a couple of days response, and the titer is huge in response to the initial thing. So this is the advantage. This is the advantage of the antibody system. Okay, this is the advantage of the antibody system, is the fact that on second exposure, we get a tremendous response to the antigen, and so that we are able to put on a good, we're able to, all things being equal, to overcome that, whatever that particular antigen is, because we produce antibodies against it, which will result in the things that we talked about: neutralization, glutination, and deactivation, and phagocytosis, and complement activation. So this is the advantage of this antigen-antibody reaction. Don't forget that these are not independent. At the same time, we could very well have a cellular response going on. Okay, secondary response, more intense, more sensitive to the antigens, stay around. They also stay around longer. Okay, these also, the antibodies stay around longer, because if you got an active infection, you want to make sure that you're getting rid of the active infection. Types of immunity. This used to be a question on the test. Natural active immunity, full-blown response to pathogen resulting in memory cells. This would be, you got the COVID-19 virus and you recovered from the COVID-19 virus. Okay. You didn't get vaccinated for whatever reason. Okay, your parents decided you weren't going to get vaccinated. Or remember this. Okay, I caution all my students in the last 4, 5, 6, 7, 8 years that we have people coming into our country, okay, that did not have the same access to medical care that you and I did. Okay, that are coming in our country, did not have access to the same medical care you did, didn't have immunizations. Okay. Didn't have regular follow-ups and things like that. So you, not me, but you are going to see diseases that I only read about. Okay, you're going to see diseases that I only read about. And so always remember that. Always be prepared to keep an open mind, because you could very well see mumps, you could very well see measles, you could very well see polio, you could very well see whooping cough. You can see things that I only read about that didn't know about. But if you get the disease, okay, if you get the disease, you're exposed to the disease, then you get memory cells. Okay, natural passive transfer of IgG and IgA antibodies from the mom to the baby. Okay, so this is natural passive immunity. Okay, this would be, if you think about it, this is a plasma transfer from a patient who had COVID-19 virus, okay, and has developed the antibodies, and we're giving it to a patient who has the active disease. So this would be natural passive, okay. Natural active, you got the disease, you survived the disease, and it doesn't have to be COVID-19. It could be anything. Okay, it could be anything. We have viruses, we have everything, all bacteria, we have all kinds of stuff around us. Most of the time, our immune system takes care of it, and we never get sick. But this is natural passive. So a second example of that would be the plasma transfer from one individual to another. Okay. Normally, we in the past, all we did was talk about transfer IgG and IgA antibodies from the mom to the baby. Okay. Artificial active is the immunization system. Okay. You were exposed to the disease by way of a immunization, but you either were given the deactivated pathogen, so you were either given a deactivated COVID-19 virus, okay, or you were given the antigen, okay, and that's in the vaccine, and you had the memory response, okay, or artificial passive intravenous injection of immunoglobulins to boost person's own response, usually produced in the lab, okay. So at some point, once we can determine what the antibody is that's effective against COVID-19. Once we determine the antibody that is effective against COVID-19, then we probably are going to be able to make this stuff. Okay. If you don't know this, vaccines as a general rule take a minimum of 10 years to produce. Okay. Vaccines as a general rule take a minimum of 10 years to produce because there are so many steps that you have to go through to produce. We are skipping a whole bunch of steps and abbreviating a whole bunch of steps, okay, to try to produce a vaccine in a year or two. But normally a vaccine takes a minimum of 10 years to produce. A vaccine takes a minimum of 10 years to produce. The flu vaccine that we get now, I think I've told you this, but if I haven't, is a group of clinicians get together and decide based on what's going on in the world, the most common flus, the most common viruses related to the flu that are going to be associated in the next flu season, and those are put together in a vaccine. So each year they should be different. Okay, each year they should be different. So probably won't be this fall, but possibly next year sometime we'll have a vaccine against COVID-19. So this is a schematic about artificial, active, and passive immunity. Like I say, this used to be a question that I asked on the test, and you'll probably get a multiple choice or true/false question on it, or matching questions. It'd be a great matching question because you'd have 4 different scenarios, and you just match it up to what was going on. Okay. Cancer and immunity, we have cancer cells going around all the time. Just most of the time we don't have an overload of cancer cells. Okay. Notice if we don't have cancer, if we just have a single cancer cell or a few cancer cells, we can take care of those. It's when we have a tumor that's shedding cells, millions of cells in a short period of time that will overcome our immune system. Immune system keeps up. Some tumors release antigens into the blood. These are some of the markers. PSA. After you get older, one of the tests they usually get on you is a PSA. That is an indication of growth in the prostate, so it may indicate that you have prostatic cancer. The CEA is a test for people with possibility of colon cancer. What you do, say you had prostate cancer or colon cancer, you had high levels of these, you had surgery, the tumor was removed, the levels should go down, and then once a year, there again, I'm not a hematologist or oncologist, but periodically you would come back and these things would be tested and they would go back to a lower level. As long as they stayed at that lower level, that would be a good thing. But if you saw that PSA going back up, then you would worry that you might have some type of metastasis or something, or you didn't get, if you did a near-term, you might not have gotten all the prostate out and you have more disease, or you might have something going on with the colon, okay? So these are good things for detection, okay? And these are good surveillance tests. Hemostatic imbalances, When I was in training, this AIDS was just coming out. When I was in training, this was the big disease, okay? Human immunodeficiency disease and AIDS, acquired immunodeficiency syndrome, okay? So this is a disease and this is a syndrome, okay? It is a retrovirus, it's an RNA virus. It infects the CD4 cells, the helper T cells. Well, why is that important? Because helper T cells are responsible for helping on the cellular immunity side and on the humoral immunity side. So they're very important. People who have this disease, if you listen to their conversations, they not only get their white blood cell counts done, but they get their particular, so they will know the number of CD4 cells and the number of CD8 cells, especially CD4 cells, okay? So they get differentiated into their white blood count gets differentiated into the number of these cells, okay? There's a glycoprotein in the HIV virus that fits the receptor CD4 cell perfectly. It is inserted into the cell. It opens up the DNA strand and it inserts itself into the host DNA. And as you're well aware, if you know about HIV, it can lay dormant for years. It can lay dormant for years. If you know about our DNA, if you remember, we talked about in Anatomy and Physiology 1, there's sections of the DNA that don't really do anything, okay? And those things get spliced out. We actually have viral DNA. Every one of us has viral DNA in our DNA that has been incorporated, but it is non-pathological. You know, we are surrounded by viruses. We have viruses in our body. Okay, we have beneficial viruses and we have non-beneficial viruses, but we have viruses in our body at this very moment. We are surrounded by viruses, just like we're surrounded by bacteria. We have viral DNA in our DNA strands, okay, which would be a nonsensical DNA for incorporation in making proteins and would be spliced out. But that has done the same thing that this HIV virus did, but it actually made a copy of itself using the RNA template and inserted it into the DNA strand. And host and replication new virus parts that infect other T cells. Eventually, the immune system gets so far behind that a secondary opportunistic infection can take hold. And so it's not the HIV virus, it's not the HIV per se that kills them, it's the fact that they're not able to mount a immune response to a normal thing that you and I would say, you know, we get a fever and chills for a day, or we'd be sick for a week, or it might not even be sick. They're not able to mount responses to this, and these opportunistic infections, okay, are usually what they succumb to. We can get hypersensitive type reactions. This is what I was talking about. An allergic reaction can cause anaphylactic shock, bronchoconstriction, and systemic vasodilatation. So you're not able to breathe and your blood pressure goes down. You get systemic vasodilatation. In other words, the vessels that have the smooth muscle that are able to dilate, all dilate up. All of a sudden, you have a huge intravascular volume, which some parts of it were shut down, and all of a sudden everything's open, and you still had the same volume of blood that you had, the same volume of fluid you had, and so now your pressure's going down, and you're getting bronchoconstriction, so you can't breathe. So this is a severe thing, okay, severe thing. So people have allergic reactions. If they're allergic to bee stings, they have an EpiPen, and what an EpiPen does, it increases the blood pressure and relieves this bronchoconstriction. And so if they're really hypersensitive to it, they have that epinephrine pen available to them. And so this is the IgE that we talked about. These are the IgEs that attach to the mast cells, okay? They attach to the mast cells. And if this particular antigen, which if it was a bee sting, okay, which was a bee sting, they get exposed to the antigen again, then all of a sudden these mast cells open up and dump out this histamine into the system. And that's what causes that, okay? Hypersensitivity type 4, that is our TB skin test, okay? These are a more localized reaction. They're usually confined to a particular area of skin. and metal poisoning or TB skin test. Autoimmune diseases, we do have autoimmune diseases. The two most common types are Crohn's disease, it's a GI system disease, and multiple sclerosis, okay? These are immune system diseases. There's more than that, okay? There's many more than that. And usually in immune diseases, what's one called Hashimoto's thyroiditis, where the immune system doesn't recognize your own thyroid as self and starts attacking your own thyroid. And so in these systems, it's the same type of thing. So we can get autoimmune diseases. They're very bad diseases to have, usually treated with, sometimes treated with steroids. And so that steroids cause their own problems. They may lessen the immune response, but if you lessen the immune response, that sets you up to a whole other set of things, okay? That's the end of the slides. I'm going to go back. If you weren't here at first, I am looking at, we just had, I will starting today and tomorrow. By Tuesday, I'll have exam 1 graded. I may unpost exam 1 so that I can grade them all and put all the scores in, then post it back up so everybody's score. As soon as I get the scores posted, I will notify you the scores are posted. All I have to do is go through and grade the short answer questions. I was looking at exam 2, which is right here, okay? And next week we did the respiratory system, so the following Monday it was going to open up on 6:15 and close on 6:22. Because this is so bunched together, I'm probably going to move exam 2 down to the next week and open it up on 6:22 and be due on 6:29. To give us a little bit of breather, because our next exam is not until July the 20th, so there's really no reason for this one to be so close next to each other. So I'm just going to move it down a week. The Pearson assignments will stay the same, because I've noticed that probably a third of the class doesn't do their Pearson assignments at the last minute, okay? So the Pearson assignments date are going to stay the same. They'll be due essentially when the exam opens, okay? I won't change those. But the exam, I'm going to move down a week, okay? The exam I'll move down a week. So you have another week to study for exam, and so you'll have a week to get the respiratory system down under your belt. This exam will only be 3 chapters, the 2 chapters on lymphatics and the immune system, and then the respiratory system. Otherwise, I don't have anything. Anybody have any questions?
Professor, I got a question.
Okay.
So for memory cells, over X amount of time, do they deplete? Like, say if you have blood loss or donate blood, like, do you lose memory cells, or will they continue to remake themselves?
They'll always be there. They'll always be there.
So your titer levels will always remain the same.
Your memory cells will always be around. Okay. So
okay.
I don't know about titer levels. That's outside of my pay grade. I could look that up, but you will always have what now it will become low, because if you step on a nail, say you have your tetanus shot, okay?
Correct.
Sometimes, and so I had a tetanus shot when I was young. Then one of the summers I worked at my dad's company, and I stepped on a nail, and they gave me a booster shot. Okay, so they reboosted my immunity and also gave me an immunoglobulin shot since I had stepped on a nail. So they are recommending, and actually that's a good question, because I never thought about that, but that in older individuals, especially older individuals that are around children, okay, say you're grandma and grandpa and you take care of the grandkids, okay, or you are a teacher and you teach in some of the grades, they are recommending booster immunizations. And so that's a great question. I never really thought about that to now. So you still have memory cells there, but the titers may be so low that maybe, and I'm just like I say, I don't have any current knowledge of this.
Right.
It may take a longer time. So that is actually a great question. Okay.
All right. Thank you.
I'm going to be 70 very soon, but if I don't forget that, I'll look that up. But I do know that they are recommended that some of the older people, especially if they're taking care of your grandkids, that you get booster immunization shots. Okay, and that's a great question.
Right.
Thanks for asking.
Thank you. No problem. Thank you.
Any other questions? All right, always email me. I prefer you email me through the JAPHILLI@FSCJ. The only reason that comes right to my phone and I see a notification right then, I have to go to the Canvas app just like you do, and I have to click on it, and then it shows me if I have any mail. I don't have any notifications from the Canvas app. So if you got really something that needs to be taken care of right then, send it directly to me on the JAPHILLI. And please don't forget, just put, you don't have to put your name on the thing, but do put BSC2086C, okay? Just put that in the subject line, because then if you say, I'm having problems with the lab for this week, I know exactly which lab you're talking about. Because I'm not with you and can see you, I don't know everybody's name who's in which class, okay? So the first thing I have to do, if you don't do that, is I have to go to the class roster in both classes and find out which class you're in. So if you don't want to put your name in the subject line, that's fine. But do put BSC2086C there, so I at least know which class you're in, so I know immediately what you're talking about, okay? That's all I got to say. And if you got something that you really need me to take care of, send it to the JAPHILLI@FSCJ, because I get a notification on my phone and I'll see that right away, okay? Otherwise, I will send out my announcement on Monday, and I will have all your tests, barring anything, barring a major storm or anything like that. I'll have all your tests ready by Tuesday, hopefully by Monday, and I'll send the notification out as soon as I get ready. Okay? Thank you very much, and we'll see you next week.